Hello everybody. Today we are going to start muscle physiology. So three types of muscle: skeletal muscle, cardiac muscle and smooth muscle. First we are going to start with the skeletal muscle followed by smooth muscle. Cardiac muscle will be discussed in cardiovascular system. So starting with skeletal muscle. If you look into any skeletal muscle of your body, like for example, suppose I am entering inside biceps muscle. So this is biceps muscle. I'm taking a small part of this biceps muscle. So I'm taking a small part of this biceps muscle. What I'm going to find out inside this biceps muscle, there will be multiple bundles of muscle fiber, and these multiple bundles are known as fascicle, right? So fascicles are nothing but bundles of fiber, muscle fiber. So if you enter inside a fascicle, you will be finding out multiple muscle fiber only. So these are the muscle fiber. If you take out this muscle fiber are nothing but a single muscle cell. So basically the fascicle is composed of multiple muscle fibers. These muscle fibers are nothing but individual muscle cells. Now if you enter inside a single muscle cell, so now you are entering inside a muscle cell, what you are finding out? Again you are finding out multiple fiber-like molecule inside the cytoplasm of the muscle cells, and these are known as the myofibril. So muscle fiber is composed of multiple myofibrils, and inside this myofibril, if you look at what you are finding out, that there is thick filament, that is the myosin, and surrounding this thick filament there are 6 actin molecule present in an hexagonal pattern. So you are finding out here these actins are present in an hexagonal pattern, and if you count them, one myosin or one thick filament is surrounded by 6 actin molecules. So myofibril is composed of your contractile protein molecules. So it is composed of contractile protein molecule, actin, myosin and others. So we all know the skeletal muscle is a striated muscle, and that is due to presence of this actin and myosin in a regular fashions. So where this actin and myosin are present, basically this actin and myosin, they are present inside this myofibril, and multiple myofibril is present inside a single muscle fiber or muscle cell. Multiple single muscle fiber is forming a fascicle, and multiple fascicle is forming a whole skeletal muscle. So this is the structural hierarchy of the skeletal muscle system, right? Fascicles, fiber and then myofibril. So now we are going to concentrate on a single muscle cell or single muscle fiber, how it looks like, what is the structural part of this cell, right? Okay, so we are going to discuss that. So first let me draw this single muscle cell. So skeletal muscle cell, it's a longitudinal cell, all of you know. So I am drawing this skeletal muscle cell. Okay, so this is a skeletal muscle cell, longitudinal in nature. So the membrane, the plasma membrane, which is surrounding the skeletal muscle cell, this is the plasma membrane, this is known as sarcolemma. So first structural part of this cell is nothing but sarcolemma. Sarcolemma. This is nothing but a normal plasma membrane of skeletal muscle cell. Now all of you know, I hope, that this sarcolemma at certain places, it will invaginate inside the cytoplasm of the cell. So the sarcolemma is entering inside the cytoplasm, and it is giving a tubular structure. This tubular structure is known as the T-tubules. T-tubules, right? T-tubules, T stands for transverse. Okay, so these tubules are placed transversely in the length of the muscle. So that's why, because of the transverse arrangement of this tubule, they are known as T or transverse tubules. In between these T tubules, there is another tubular structure. Okay, so now look at this green color structure. Okay, it is present longitudinally along the length of the muscle with two terminal spherical region. So this is terminal spherical region with a longitudinal portion and again a terminal spherical region. This tubule, it is present longitudinally along the length of the muscle. That's why this is known as L tubules. L stands for longitudinal. So this is L tubule. All of you know that this L tubule is nothing but your sarcoplasmic reticulum, and all of you know sarcoplasmic reticulum is nothing but storage house of calcium inside the cell, although the main calcium is stored inside the terminal portion of this sarcoplasmic reticulum. So the main calcium is stored inside this terminal portion, and this terminal portion of sarcoplasmic reticulum is known as cisterna. So basically, cisternal region are the main region where calcium is stored. Now the combination of these T tubules and L tubules, so T tubule and L tubule, combination of these two structures are known as sarcotubular system. So second layer of structure is sarcotubular system. Sarcotubular system. Okay, right. And the third level of structure, if you enter further inside the cytoplasm of this muscle cell, okay, inside the cytoplasm you are going to find out multiple fiber like molecule. These fibers are nothing but your myofibrils. So these are the myofibril, okay, multiple fiber like molecule present inside the cytoplasm of this muscle. These are my myofibril. And I told you earlier that it is the myofibril which contain dark band and light band. So this is dark band followed by light, again dark band followed by light. So this kind of band will be present inside the myofibril because myofibril contain actin, myosin and other contractile elements of the muscle. Getting my point? So it is the myofibril which is giving the dark and light appearance of the skeletal muscle, and that's why it is known as striated muscles. And last point is that there will be presence of multiple nuclei inside a single cell. So skeletal muscle is a multinucleated cell, multiple nucleus will also be present. So if I write here, the third layer of structure in skeletal muscle cell is nothing but myofibril, right? Myofibril. So skeletal muscle cell, structurally it is composed of three layers of structure: sarcolemma or plasma membrane, sarcotubular system or T tubules and L tubules, and the myofibril within which the contractile elements are located. Now the sarcolemma, it's a normal plasma membrane, so obviously it will be composed of lipid molecule as well as protein molecule, as we have discussed earlier in general physiology. But the protein, okay, special types of proteins are present in sarcolemmal membrane, and that you have to remember. So we all know the proteins which are present in membrane, they are of two types: integral membrane protein and the peripheral membrane protein. Integral and peripheral membrane protein. The special integral membrane protein which is present in skeletal muscle membrane is known as dystroglycan and sarcoglycan. sarcoglycan. Dystroglycan and sarcoglycan, they are the integral membrane protein specially for skeletal muscle cell membrane or sarcolemmal membrane. And the peripheral membrane protein, again two special protein we have to remember, that is dystrophin and syntropin. syntropin, right? Please don't forget this name because MCQ already is asking about this protein. So if I understand here, so this is the sarcolemma membrane, this is the sarcolemma or the plasma membrane of the skeletal muscle cell. Integral protein, they will be present through and through the cell membrane. Suppose this is dystroglycan, and along with dystroglycan, the sarcoglycan complex will also be there. So this is the sarcoglycan complex, dystro and sarco. Now along with this dystroglycan, a long protein molecule is attached, and this protein is a peripheral protein. It is nothing but dystrophin, and along with this dystrophin, small peripheral protein will also be present. This is nothing but syntrophin. So what these membrane proteins are doing here, this membrane protein, okay, extracellularly, they remain attached with the extracellular matrix protein like laminin. And intracellularly, this protein will remain attached with cell cytoskeleton protein, particularly the actin cytoskeleton protein. By this kind of attachment, so it is attached outside also, it is attached inside also. By this kind of attachment, it is giving tensile strength to the membrane, it is giving strength to the membrane. That's why we can easily understand that if there is mutation of any of these membrane protein, muscle will be weak, myopathies will develop. So we'll be discussing that. So look at this membrane protein first in diagram which is taken from genome. Right. So this is sarcolemmal protein that we have just discussed. You can find out here dystroglycan protein, beta dystroglycan and alpha dystroglycan, right? And you can also find out the sarcoglycan complex. So this dystroglycan complex and the sarcoglycan complex, they are the integral membrane protein. Along with that, you can find out this long blue color protein. This is nothing but your dystrophin, and along with dystrophin, the syntrophin is also attached. I told you these proteins remain attached with extracellular matrix laminin component and intracellular actin component. So this is nothing but your actin component, that is the cell cytoskeleton component. By this way, it is giving strength to the membrane. And that's why mutations of this protein, particularly the dystrophin mutation, all of you know that if the dystrophin mutation is so severe that the protein is completely absent from muscle cell, then a severe form of disease originate, that is known as Duchenne's muscular dystrophy. And if this dystrophin protein is partially present or partially mutated, the dystrophin protein will be reduced in number inside the cell. So less severe form of disease will be there, and that is known as Becker's muscular dystrophy. Along with that, if there is mutation of this sarcoglycan complex, then another form of muscular dystrophy originate, that is known as limb-girdle muscular dystrophy, right? So please remember all these muscular dystrophy, they are very important. I told you already that this Duchenne's muscular dystrophy and this Becker's muscular dystrophy, both of them are dystrophin mutation, but Duchenne's is severe form of disease, the protein is completely absent, and Becker is a milder form of disease where the protein is present but reduced in number or maybe altered in quantity. Because of this, the muscles will be affected, particularly the lower limb muscle will be affected first, and progressively it will ascend to the upper part of the body. There will be pseudo hypertrophy of multiple muscles, particularly this cup muscle pseudo hypertrophy is a very prominent feature of this disease, and due to involvement of the heart and other organ, the death occurs by 30 to 40 years of age. This is very severe form of disease, that is the Duchenne's muscular dystrophy. Death occurs by 30 to 40 years. And Becker is a milder form of disease. I told you also that this limb-girdle muscular dystrophy, that is due to mutation of the sarcoglycan complex and other component of the dystrophin protein. From the name you can understand the limb-girdle means the muscles that are going to be affected in and around the shoulder region and in and around the hip and leg region muscles. So this is the group of muscles which are going to be affected in limb-girdle muscular dystrophy. So all this disorder are particularly related to sarcolemmal membrane protein that we have discussed here, that is dystrophin and sarcoglycan complex, right? So first is done. Okay, so sarcolemma is done. Okay, now the next thing is that sarcotubular system. So sarcotubular system, we already discussed that it is composed of T tubules T tubules and it is composed of L tubules. L tubules. T tubules is nothing but a part of the cell membrane only. So this is nothing but sarcolemmal membrane only. And L tubules, it is nothing but sarcoplasmic reticulum. Right? Sarcoplasmic reticulum. Now, very, very important. T tubules, okay, they contain a channels, calcium channel, and that channel is known as DHPR type of channel. So what is this DHPR? You know from your pharmacology knowledge that it is dihydropyridine receptor channel. Dihydropyridine receptor channel. What is this? This is a basically L-type calcium channel. This is a basically L-type calcium channel which is obviously voltage gated. So voltage gated L-type calcium channel is nothing but DHPR, dihydropyridine receptor channel. This is the same channel which is blocked by different calcium channel blocker. Okay, this same channel is also present in vascular smooth muscle. Okay, and it is blocked by certain groups of calcium channel blocker. That's why this is known as dihydropyridine calcium channel. Right. So dihydropyridine receptor channel, it is nothing but a L-type calcium channel. The recent name of this channel, okay, is that nowadays the channel is known as CA.V 1.1 type of channel. What I'm saying? So I told you that DHPR channel, it is nothing but a voltage gated calcium channel. How it is written in recent nomenclature? So CA, that is calcium, V stands for voltage gated, and is subtype 1.1. DHPR channel subtype is 1.1, which is present in skeletal muscle, right? I'll be coming in detail of this different subtype, but hold on. Okay, so on the other hand, the L tubules contain another calcium channel that is known as RYR, ryanodine type of channel, ryanodine receptor channel. All of you know that this channel is blocked by ryanodine groups of alkaloid. That's why this is known as ryanodine receptor channel, which is known as RYR channel. This RYR channel, they are particularly present at the level of the cisterna. Now look at this diagram. So this is the normal skeletal muscle cell. On the T tubular membrane, there will be voltage gated calcium channel, which will be known as DHPR channel, and just opposite to this, the cisternal region will contain another calcium channel which is known as RYR type of calcium channel, right? RYR type of calcium channel. Now I told you that DHPR, look at here, the DHPR is also known as CAV 1.1. So what is the different subtypes or different name of this? Okay, you please remember, this is very, very important. CAV 1.1, this is present in skeletal muscle. Okay, skeletal muscle. So this is L type of voltage gated calcium channel, but for skeletal muscle, this is CAV 1.1. CAV 1.2 is present both in the myocardium, it is also present at the level of smooth muscle, right? So this is different subvariety of L type voltage gated calcium channel. Now this ryanodine receptor channel, which I told you earlier, RYR, it also has three subvariety. RYR1 is particularly for skeletal muscle cell, RYR2, it is particularly for myocardium cell, and RYR3, this is for smooth muscle cell. It is for smooth muscle cell. So please remember, this is very, very essential. Okay, so voltage gated calcium channel, they are different types. L type voltage gated calcium channel nowadays is known as CAV 1.1 or 1.2. 1.1 is particularly present for skeletal muscle, 1.2 is particularly present for myocardium and smooth muscle. Now if I give you the detailed list of this voltage gated calcium channel, you will be finding out here. Just hold on here. Okay, so RYR1, skeletal muscle, 2 myocardium, 3 smooth muscle and epithelium cell. Okay, on the other way, here all of you know that voltage gated calcium channel, it may be of L type, PQ and N type and T type voltage gated calcium channel. L type voltage gated calcium channel, as you can find out here, they are also known as DHPR type of channel, dihydropyridine receptor channel. 1.1 is dedicated for skeletal muscle, 1.2 is dedicated for myocardium and smooth muscle. Now, what about this PQ and N type? PQ type is also known as CAV 2.1 and N type calcium channel is also known as CAV 2.2. Getting my point? So PQ and N type of calcium channel, this is basically 2.1 or 2.2. L type calcium channel is basically 1.1 or 1.2. And this T type calcium channel, they are 3.1, 3.2 or maybe 3.3. This way you have to remember that different types of calcium channel. T type calcium channel is particularly present in the heart muscle, the pacemaker tissue and osteocytes. Right? So this way this classification may be very, very crucial for future MCQ. Try to remember this. So now we are discussing the sarcotubular system. Where? Here. Okay. So we are discussing this sarcotubular system, T tubules and L tubules, and we have seen different types of channels are present there. How these T tubules and L tubules, how they are functionally connected. This is known as excitation contraction coupling of muscle. So our next topic is excitation. Excitation contraction coupling. We are going to discuss this excitation contraction coupling of skeletal muscle and cardiac muscle parallelly. Right? So I am drawing the skeletal muscle membrane. Okay, it is forming the T tubules, right? So this is the sarcolemma and T tubule. I hope all of you know this. Okay, and this is the cisterna. So this is sarcoplasmic reticulum. There will be another cisterna on the other side of the T tubules. So this is another sarcoplasmic reticulum, right? So this is membrane, that is sarcolemma. Which muscle I am drawing here? This is nothing but the skeletal muscle, right? Skeletal muscle. And parallelly I am also going to draw the myocardium. So similar kind of picture, the muscle membrane, okay, forming T tubules, okay, and there will be cisterna and sarcoplasmic reticulum, right? So this is typical of myocardium. Okay, myocardium, and here is your skeletal muscle. Now first difference you remember that in skeletal muscle, one T tubules is surrounded by two cisterna on both sides. One T tubule surrounded by two cisterna on both sides. So it is number 1, this is number 2 and this is number 3. What I am saying? It is making a triadic junction. So please note down, the sarcotubular system in skeletal muscle, they form a triad. That means this is one cisterna, okay, then number 2 is the T tubules and number 3 is another cisterna. So 1, 2, 3, it is forming a triadic junction. That's why we say that in case of skeletal muscle, it's a triadic junction between L and T tubule. Okay, so these L tubules and these T tubules, they are forming a triadic junction in skeletal muscle. In case of cardiac muscle, if you look at the diagram, then one T tubule is surrounded by only one cisterna on one side. The other side cisterna will not be present in case of myocardium. So in case of myocardium, one T tubules and one cisterna. That means it is forming a dyadic junction between T and L tubule. Dyadic junction in case of myocardium. Now, This T tubule will contain the calcium channel that we have discussed earlier. So which type of calcium channel could you require? This is CAV 1.1, and just opposite to this there will be RYR channel. So this is my RYR channel, but here it is RYR1 type of channel. Am I clear? Okay. Same way, if I look at the myocardium channel, then there will be DHPR channel. This DHPR channel will be known as voltage gated calcium channel, but here it is CAV 1.2 types of channel. And just opposite to this there will be RYR channel. So this RYR type 2 type of channel will be present here. So far this is clear. Now we'll be first discussing the skeletal muscle. So whenever there is an action potential on the skeletal muscle membrane, suppose this is an action potential, okay? Action potential means there will be entry of sodium ion. So sodium ion entry means there will be positivity. So membrane will be positive, cell membrane will be positive inside. So now this positivity or this depolarization will move through the cell membrane. It is moving through the cell membrane and it is depolarizing the T tubules. Whenever the T tubules is depolarized, the DHPR channel which is present on these T tubules is going to be activated, right? So whenever there is activation of this DHPR channel, what will happen? The DHPR will change its conformation. What I'm saying? DHPR, okay, it's a protein only, it's a channel, right? So it's a protein, and just opposite to this there is RYR. Whenever the DHPR is activated by depolarization, it will change its conformation and there will be extension of one of the arm from this channel, and this arm is going to interact with the RYR. Again I am repeating. So whenever this DHPR is activated, the conformation of this DHPR will change. One arm or one part of this DHPR will be extended from here to the RYR, and they too will interact mechanically or physically. So now this DHPR is interacting with the RYR mechanically and physically, and because of this interaction, the RYR channel will be twisted a little bit. What I'm saying, whenever this DHPR channel is extending its arm and interacting with the RYR, it will cause twisting of this RYR channel, and because of this twisting, the RYR channel will open up, and because of the opening up of this RYR channel, what is going to happen? Huge amount of calcium is going to enter from cisternal region into the cytoplasm of the skeletal muscle cell. So now you look at the whole mechanism, how this is happening. So I'm writing, first there is an action potential on muscle membrane that is causing depolarization of T tubules. Because of the depolarization of the T tubules, what is happening? The DHPR channel is activated. Activation, DHPR channel activation. Now whenever this DHPR channel is active, what this DHPR is doing? DHPR is interaction with RYR type 1 channel. What kind of interaction? Mechanical interaction or physical interaction, and because of this mechanical interaction, the RYR channel will be twisted, and because of which RYR channel opening is going to happen. Because of the opening of the RYR channel, the calcium influx will occur. The calcium influx will occur within cytoplasm of this muscle, and this calcium is going to cause contraction of the skeletal muscle. This is going to cause contraction of the skeletal muscle. So now if you look at here, this DHPR and RYR interaction, this is known as coupling. They are coupled, DHPR, RYR. They are sitting very near to each other. Only 11 nanometer gap is there in between these two. But This interaction require action potential because it is the depolarization which is going to activate the T-tubules channel, that is DHPR channel, and then only they are going to interact, and this interaction is mechanical interaction. So this DHPR and RYR coupling, this is known as electromechanical coupling in skeletal muscle. This is known as electromechanical, very important, electromechanical coupling between DHPR and RYR channel. So electro means in presence of electrical activity, that is action potential is required. In presence of electrical activity, there is mechanical coupling between DHPR and RYR. This is typical of skeletal muscle and this is known as electromechanical coupling. So whenever this electromechanical coupling is there, what is happening? The calcium is released from cisternal region into the cytoplasm. This small release of calcium from cisternal region is sometimes known as calcium spark. Calcium SPARK. Sparking of calcium. Okay, you have to understand that this release of calcium is occurring from multiple sarcoplasmic reticulum which are present. So inside the skeletal muscle cell, in multiple areas there is sparking of calcium current, and that's why this is sometimes known as calcium spark. This calcium spark is also occur in myocardium also as well as in smooth muscle also. So calcium spark is nothing but release of small amount of calcium from cisterna to the cytoplasm, right? But the bottom line, we have to remember that in case of skeletal muscle, the interaction between DHPR and ROER is electromechanical interaction. Now let's understand what is happening in case of myocardium or heart muscle. Right. So again the same thing. First you require one action potential. So suppose one action potential is reaching on the surface of the membrane. Again, the action potential in myocardium, this is also based on sodium channel. So sodium is going to enter, and because of the sodium there will be progressive depolarization of this membrane, right? So this is depolarization. Now this depolarization will travel through the T-tubules, right? So it is traveling through T-tubules and it is going to activate the DHPR channel, that is CAV 1.2 types of channel. So as you can find out here, although the channel is DHPR channel, although the channel is L-type voltage gated channel, but it is a different subvariety in case of myocardium, and that's why this DHPR channel in case of myocardium, whenever it is activated by depolarization, the channel will open up itself. What I'm saying, this channel is going to open up. Okay, this opening up was not happening in case of skeletal muscle. DHPR channel was never opening in case of skeletal muscle because that is a separate subvariety. But in case of cardiac muscle, this DHPR, whenever it is activated, it is opening up itself. So if this channel is open up, it's a calcium channel. Calcium is going to enter inside the cell through this channel. Where from this calcium is coming? This calcium is coming from extracellular fluid. So this calcium is coming from extracellular fluid or blood calcium or interstitial fluid calcium. So myocardium, whenever it is activated by action potential, right, there is activation of the DHPR channel. DHPR channel activation means small amount of calcium is going to enter in the cytoplasm. Small amount of calcium is going to enter in the cytoplasm, and this calcium is coming from the extracellular fluid. But note down, okay, this calcium which we are discussing here, which has entered from extracellular fluid, the amount of this calcium is very small. So it is not going to cause full contraction of the myocardium. You require the cisternal calcium, you require the sarcoplasmic reticulum calcium. So what you are going to do? Remember that this RYR type 2 channel in case of myocardium, this is calcium gated calcium channel. Oh my god. So this is calcium gated calcium channel. What do you mean by this? Means The RYR channel, it contain gate, and that gate will open in presence of calcium. That's why this is known as calcium gated calcium channel. In case of skeletal muscle, they are mechanical interaction, so that is a mechanical channel. RYR type 1 is a mechanical channel, but here it is a calcium gated calcium channel. So the extracellular fluid calcium which has already entered inside the cell, okay, this calcium which has already entered inside the cell, now this is going to stimulate the RYR channel. Because of the stimulation, further calcium is going to enter in the cytoplasm due to opening up of RYR channel. Getting my point? So what is happening? The extracellular fluid calcium which has already entered inside the cell, this calcium is going to stimulate the RYR channel, and whenever RYR channel is stimulated, more calcium is going to enter in the cytoplasm from sarcoplasmic reticulum. That means cardiac muscle is taking help of two sources of calcium. One calcium is coming from the extracellular fluid and one calcium is coming from sarcoplasmic reticulum of the myocardium. So if I try to understand this phenomenon once again with some write-up, so action potential on myocardium cell, that is causing depolarization of T tubules. Very good. This is same to same with that of the skeletal muscle. This depolarization is causing activation of DHPR channel. DHPR channel activation means it will open up. So it will leads to opening of DHPR channel. This is very, very different from skeletal muscle. This opening was not happening in skeletal muscle. Note down, note down, note down. Now, whenever there is opening of DHPR channel, okay, ECF calcium enters inside the cell, enters inside into the cell, right? This ECF calcium is now going to stimulate the RYR type 2 channel. Okay, it is going to stimulate the RYR type 2 channel, because of which the RYR type 2 channel will open up. So it will leads to opening up of the RYR channel, due to which further calcium will enter. Okay, calcium entry into cytoplasm, right? Cytoplasm, and then it will leads to contraction of the myocardium. So I can understand here this phenomena is a different from skeletal muscle phenomena. Okay, so here I can find out that extracellular calcium is stimulating a release of further calcium from sarcoplasmic reticulum. This phenomena is known as calcium induced calcium release phenomena in cardiac muscle. Very, very important. CICR. What is this? This is calcium induced, okay, calcium release phenomena. Calcium induced calcium release phenomena. One calcium is inducing release of further calcium. So this first calcium is the ECF calcium, and the second calcium is the sarcoplasmic reticulum calcium. Calcium induced calcium release phenomena is particularly not seen in skeletal muscle. It is a dominant phenomena in case of cardiac muscle. And if I ask you what type of coupling is occurring here, DHPR and RYR, what type of coupling is occurring here? This is known as electrochemical coupling. Electrochemical coupling between, okay, DHPR and RYR channel. What do you mean by electrochemical? Electro is in presence of electrical activity, that is action potential, and chemical coupling means that DHPR and RYR, they are interacting through a chemical. And what chemical that is? That is nothing but calcium. That's why this is known as electrochemical coupling in case of cardiac muscle. Getting my point? So now if I ask you a bottom line, okay, if I ask you a bottom line that this is the skeletal muscle cell and this is the myocardium cell, we have seen the phenomena in two cells. Now you tell me your MCQ, source of calcium for skeletal muscle contraction. Source of calcium for skeletal muscle. Okay, what is the source of calcium? Where from the calcium is coming for skeletal muscle contraction? We can understand that for skeletal muscle only on one source of calcium, and that is nothing but cisternal calcium. But if I ask you what is the source of calcium for myocardium, then you have seen earlier that two sources. One source is coming from ECF and another source is coming from sarcoplasmic reticulum. So we are writing that because that is very, very important. So source of calcium for skeletal muscle contraction, 100% calcium is coming from sarcoplasmic reticulum. But if I ask you sources of calcium for myocardium, your answer will be that 80% calcium is from sarcoplasmic reticulum and small amount of calcium is coming from ECF calcium. Okay, so this 80% calcium is coming from intracellular fluid and 20% source of calcium is coming from, right, extracellular fluid. Okay, so this is very, 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 very important. But there is a very crucial question. If I ask you, in myocardium, which calcium is the essential source of calcium? Essential source means without that source, the myocardium is not going to contract. Then your answer will be, although this ECF calcium is small in amount, but this is the essential source of calcium, because we have already seen that if this ECF calcium is not entering, then this ROS channel 2 opening is also not going to happen, because I told you the mechanism. First, the ECF calcium is going to enter inside the cell, and then the ECF calcium is going to stimulate the sarcoplasmic release calcium. So ECF calcium is essential for myocardium contraction. This you also have to remember, right? So now if you look at these two sources, I can understand that in skeletal muscle 100% source of calcium is coming from sarcoplasmic reticulum, and in case of myocardium only 80% source is from sarcoplasmic reticulum. So volume of sarcoplasmic reticulum, the size of sarcoplasmic reticulum will be bigger in which cell? Obvious. So that is another difference. The size or the volume of sarcoplasmic reticulum, it is bigger in case of skeletal muscle cell than that of the myocardium, right? Then second, size of T tubules, or T tubules diameter. What do you think? These T tubules which are present in case of skeletal muscle, there is no entry of calcium through this T tubule, right? But if you look at the T tubule in myocardium, there is entry of calcium through this T tubule. So if the calcium is entering through T tubule, you have to give a larger diameter of this T tubule, otherwise calcium is not going to enter easily. So whenever I'll ask you the diameter of T tubules is large in which cell, your answer will be size or diameter is more in case of myocardium cell than in case of skeletal muscle cell. Getting my point? So this is very, very important thing that you have to remember about this DHPR and ROIR interaction. So far we have seen many different types of channel. Okay, all these channels are very, very important for your MCQ, but apart from that you also have to remember certain mutation of this channel. Okay, note down. So you know that mutation of this ROIR type 1 channel, very, very important. Mutation of this ROIR type 1 channel means the disorder is going to affect the skeletal muscle cell. Okay, if there is gain of functional mutation means because of the mutation the channel become hypersensitive, means the opening probability of the channel increases. We all know the disease is known as malignant hyperthermia. Okay, and if there is loss of functional mutation of this channel ROIR1, then the disease is known as central core diseases. diseases. So if there is gain of functional mutation, I can understand that through this ROER type 1 channel, huge amount of calcium will be enter in the cytoplasm, and that will lead to severe type of muscle contraction with huge amount of heat production. That is a typical feature of malignant hyperthermia: muscle stiffness, heat production, and there is hyperthermia. And if there is loss of functional mutation, I can understand that calcium which is entering in the cytoplasm, that is lesser in amount. So muscle is not going to contract strongly. So obvious symptoms of this disease, that is a central core disease, will be weakness of the muscle. So please look at this. Okay. So two disorder. One is malignant hyperthermia, right? Malignant hyperthermia, and there is central core disease. In both of these disorder, the channel which is mutated is ROIR type 1, means skeletal muscle is going to be affected. All of you know that malignant hyperthermia is precipitated by certain form of anesthetic agent. You know that certain form of anesthetic agent, like for example halothane, okay, sevoflurane, or muscle relaxant like succinylcholine. Or it is also written in books that malignant hyperthermia can be precipitated by extreme severe form of exercise or extreme temperature. Okay, so halothane and similar kind of inhalation anesthetic agent. You have to remember succinylcholine, extreme exercise, extreme heat can precipitate this malignant hyperthermia. What will happen? Whenever the disease is precipitated, ROIR channel will open up and it will not be able to close spontaneously. That is increase in open probability of this channel. Due to huge amount of calcium influx, muscle will be strongly contracted, there will be rigidity of the muscle, huge amount of temperature will develop, high fever, along with there will be increase in heart rate. Rhabdomyolysis can also happen, okay, and because of the muscle membrane disruption, potassium will come out from this muscle, potassium level in the blood will be higher. The treatment for this malignant hyperthermia, the drug which is used is dantrolene. It is said that dantrolene binds with ROIR type 1 channel and prevent its opening. So your solution is given. So dantrolene is the drug which antagonizes the ryanodine receptor channel. If you look at the central core disease, I told you the predominant symptoms is weakness. That's why there will be hypotonia, decreased muscle tone, there will be weakness. So obviously there will be mild delay in development of the children, and weakness of the facial muscle will also be seen. No specific treatment is there for central core disease. Right? So this is about ROIR channel mutation. Now further mutation, if there is mutation of this CAV1.1 type of channel mutation. Okay, so calcium channel CAV1.1, this is mutated in a disease which is known as hypokalemic periodic paralysis. Periodic paralysis. Okay, so you must have heard of this disease, hypokalemic periodic paralysis. There is transient, sudden onset paralysis or muscle weakness can develop associated with hypokalemia. So whenever your potassium level in the blood is very less, sometimes the patient suffer from severe form of muscle weakness or maybe mild form of muscle weakness or paralysis, that is known as hypokalemic periodic paralysis. The channel which is getting mutated in this disorder is CAV1.1. So this is a calcium channel mutation disorder, hypokalemic periodic paralysis. But if you look at the hyperkalemic periodic paralysis, hyperkalemic periodic paralysis, again similar form of periodic paralysis, okay, but this disorder occurs whenever there is blood calcium is very high. This is due to sodium channel mutation, and that is voltage gated sodium channel Na.V 1.4 type of voltage gated sodium channel mutation. So now you are getting confused, what is this NAV 1.4? So NAV 1.4 type of voltage gated sodium channel. We have discussed voltage gated sodium channel in neuron, that the action potential of the neuron is based on voltage gated sodium channel. Same voltage gated sodium channel is also responsible for action potential generation on myocardium as well as on your skeletal muscle. So neuron, skeletal muscle, myocardium, all three voltage gated sodium channel. NAV is the voltage gated sodium channel. Now this voltage gated sodium channel is composed of two subunits, alpha and beta. Depending on the alpha subunit, there are different variety of this voltage gated sodium channel starting from NAV 1.1, 1.2 .1.9, means total 9 different sub variety of voltage gated sodium channel is there, that is NAV 1.1, NAV 1.2, NAV 1.3 up to NAV 1.9. The particular voltage gated sodium channel which is present in case of skeletal muscle, note down, this is important, NAV 1.4. The particular voltage gated sodium channel which is present in case of myocardium, this is also fixed, NAV 1.5, and all other starting from 1.1 or 1.2 or 1.3 or 1.6, 1.7, 1.8 or 1.9. All of these are neuronal voltage gated sodium channel. Getting my point? So multiple many channels, many subvariety we are talking about in this class. Please try to remember. Okay, so for skeletal muscle this is fixed 1.4, for myocardium this is also fixed 1.5, for neuronal all others type of sodium channels are present. So I told you that hyperkalemic periodic paralysis is due to mutation of NAV 1.4 subtype, that is the skeletal muscle voltage gated sodium channel, right? So if I go back to this diagram once again, so this is the diagram, okay, excitation contraction coupling diagram, skeletal muscle. I told you action potential is based on sodium channel, voltage gated sodium channel. If I ask you what type of voltage gated sodium channel is here, your answer is NAV 1.4. Same way, if I ask you in case of myocardium, again this is a voltage gated sodium channel, NAV, this is subtype 1.5. And if this NAV 1.4 is mutated, that is in case of skeletal muscle, then the disease is known as hyperkalemic periodic paralysis. And if this CAV 1.1 is mutated, then the disease is known as hypokalemic periodic paralysis. And if this RYR1 is mutated, then it can be of malignant hyperthermia also, or can be of central core disease also, right? So this is all about the muscle contraction, excitation, contraction, coupling mechanism and different types of mutation. Now, whenever we are discussing that muscle contraction is based on this calcium, the question arises that what about relaxation? How this calcium is going to enter inside the sarcoplasmic reticulum once again? How about relaxation? So note down, in case of skeletal muscle, this calcium will be mopped inside the sarcoplasmic reticulum once again by a pump, and that is known as SERCA, SERCA. SERCA, SERCA. If I look at here, A stands for sarcoplasmic, ER stands for endoplasmic reticulum, C stands for calcium, A stands for ATPase. बहुत ज़्यादा हो गया. So please note down. So this is sarcoplasmic, endoplasmic reticulum, calcium, ATPase molecule. So this is basically a primary active transporter. Pump, sarcoplasmic, endoplasmic reticulum, means it is present in the sarcoplasmic reticulum membrane also present in the endoplasmic reticulum membrane, and it's a calcium ATPase molecule, primary active transporter. So whatever calcium has entered in the cytoplasm from sarcoplasmic reticulum, that is taken up once again inside the sarcoplasmic reticulum by this SERCA pump, and this will cause relaxation of the skeletal muscle, right? But in case of cardiac muscle, if you look at, in case of cardiac muscle, you are finding out that cardiac muscle was getting two sources of calcium. One calcium was coming from extracellular fluid and one calcium was coming from sarcoplasmic reticulum. So this is the two source of calcium, as you can find out here. So this is one source which is coming from extracellular fluid, and this is another source that is coming from sarcoplasmic reticulum. So I have to remove this green color calcium out of this cell because it is the extracellular calcium. So I am returning back this extracellular calcium once again with the help of an exchanger which is throwing calcium in exchange with sodium, and this is typical of NCX, sodium calcium exchanger. We have discussed this exchanger in general physiology chapter. So it is a secondary active transporter, sodium calcium exchanger, and the calcium, okay, which has entered from sarcoplasmic reticulum, I am giving back to the sarcoplasmic reticulum once again with the help of SERCA pump, SERCA pump, right? So now if we write down this thing, okay, here, suppose I'm writing down relaxation of the muscle, relaxation of muscle. So in case of skeletal muscle, the relaxation is completely done by SERCA pump, okay, and this is an primary active transporter. In case of cardiac muscle, the relaxation is done by SERCA pump also, as well as NCX pump also. NCX is an secondary active transporter and SERCA is an primary active transporter. Getting my point? Because in cardiac muscle, it was getting calcium from two sources. One is from extracellular fluid and one is from intracellular fluid. But skeletal muscle was getting calcium from only one sources, right? That's why SERCA pump is enough for complete relaxation of the skeletal muscle. This SERCA pump, okay, is a protein molecule which is produced by genes which is known as ATP2A1. This is the gene which causes synthesis of SERCA pump protein. This gene sometimes can get mutated. So mutation of this gene, which is generating the SERCA pump protein, that is ATP2A1 gene, if this is mutated, the disease is known as BRODY, Brody's myopathies. Getting my point? So what will be the symptoms of this Brody's myopathies? Could you guess? So SERCA pump is required for contraction or relaxation? Relaxation. So if this SERCA pump is mutated, the relaxation of the skeletal muscle will not be proper. So in case of Brody's myopathy, the typical symptoms is that severe muscle stiffening or muscle cramping after exercise or after activity of the muscle. So when the muscle is already contracted, muscle is doing activity, but it is not relaxing properly, what will happen? There will be severe muscle stiffening, severe muscle contraction, okay, and severe muscle cramp. That is the symptoms of Brody's myopathy, right? So contraction, relaxation, everything is done. Now the last thing before concluding this chapter, I am going to talk about another protein, which is the protein present inside the sarcoplasmic reticulum. So this is a bigger version of sarcoplasmic reticulum, okay, and here is your RYR channel. There are certain protein present inside the sarcoplasmic reticulum, okay. They are known as calsequestrin, QUESTRIN, okay. This calsequestrin, and second protein is calreticulin, okay. Remember, calsequestrin protein is present inside the skeletal muscle also, inside the myocardium also, as well as inside the smooth muscle also. But this calreticulin, this is present only in case of smooth muscle sarcoplasmic reticulum. So all this protein, calsequestrin and calreticulin, right? Calsequestrin and calreticulin, they are protein which are present inside the sarcoplasmic reticulum. What they are doing? These are the calcium binding protein. What do you mean by that? Means just now I talked about that this SERCA pump, they sarcopump, they are mopping up calcium from cytoplasm inside the sarcoplasmic reticulum. Now, inside this, if the calcium concentration is very high inside the sarcoplasmic reticulum, if the calcium concentration is very high, then this activity of the sarcopump will be decreased. So what I have to do? I have to reduce this free calcium concentration inside the sarcoplasmic reticulum. And this calciquestrin and calreticulin, they will bind with calcium and it will convert the free calcium into a bound form of calcium. That means I can say that this calciquestrin and calreticulin, they are the calcium chelating agent. They are decreasing the free calcium concentration within sarcoplasmic reticulum. Along with that, calciquestrin also regulate the opening and closing of the RYR channel. Calciquestrin is also a regulator of RYR channel. So this is all about the first part of skeletal muscle.